Hey, welcome back. I'm going to start the Chapter 24 Urinary System lecture. This is a short lecture. It's only 4 pages, so hopefully we'll be able to get through it pretty quickly. We're going to talk about briefly how the urinary system works. So we'd already talked about the kidneys getting a large portion of cardiac output, especially during rest. So the kidneys are going to process around 180 liters of filtrate. That's how much they're going to filter per daily. However, only about 1.5 liters is actually going to be excreted as urine. So there's a whole lot that happens in between 180 liters and 1.5 liters. And remember, normal blood volume is only around 5 liters. So you're going to filter your entire blood volume approximately 60, that's the blank, times per day. Now, making urine involves 3 processes. We're going to talk about all 3. And that's going to be what we're covering in this lecture. All right, so the first one, if you want to look here, it's on page 955, figure 2410. The first process, glomerular filtration. Glomerular filtration, it is passive, it is not selective, and this is where fluid passes from the glomerulus, which is the blood vessels, so from the circulatory system, to Bowman's capsule, which is the beginning of the nephron. Remember, we talked about the nephron, functional unit of the kidney, in lab. And that happens as a result of hydrostatic pressure. So pressure pushes the fluid from the bloodstream into the nephron. There's a highly permeable membrane that's going to separate the blood from the inside of the Bowman's capsule, part of the nephron. The second part, tubular reabsorption. This is what's happening. We are fully in the nephron now. Substances are reabsorbed from the tubules in the nephron. They're going back into the capillaries. So things that are reabsorbed do not appear in the urine. They're things we want to keep. There is a process also called tubular secretion. This is where substances pass from the capillaries around the nephron back into the filtrate, and they are going to be in the urine. These are things you don't want. And ultimately, the fourth process is excretion, which is where you actually expel urine. So it's just a transportation process. All right, starting glomerular filtration. Page 955. The filtration membrane, it's going to be between the blood, so that's going to be blood vessels within the glomerular capillary, and the interior of Bowman's capsule. So if we're on our figure here, here's our figure, we're talking about this portion right here. Here's the blood there, and then here's the beginning of the nephron. So our filtration membrane, where we're going to be taking blood from, fluid from the capillaries into the nephron, it is very permeable because the capillaries in the glomerulus, they are fenestrated. So that means they have larger openings, fenestrated. Within the visceral membrane of the Bowman's capsule, that's going to be the inside, there are podocytes. So you're talking about cells with, the nucleus, and then the cell body is split up into various little legs or feet, which allow for filtration slits. That's the third blank, filtration slits, which are going to be the spaces between those podocytes. So the capillaries got holes in it, and then there's these slits in Bowman's capsule in that visceral membrane, so fluid can go out and into the nephron fairly easily. So it's going to allow passage of small molecules. Here's what you need to know that gets filtered: water, glucose, amino acids, and nitrogenous waste. Ultimately, we're talking about the urinary system, so we're trying to get rid of waste. But we have to filter just about everything. Now, there is space, so things are moving. So not everything goes through, not everything gets filtered. So large plasma proteins and other large things don't normally get filtered. If they get filtered, that's an indication that there is an anatomical problem. So things you don't want to see in urine: red blood cells, albumin, which is our primary plasma protein. You should never find that in the urine. You don't want to filter any sort of blood cells, white blood cells, things like that. Now, glomerular filtration rate. This is the rate that fluid is being filtered from the blood into the nephron. So normally we call it GFR. It's a measure of kidney function. It's going to be 120 to 125 milliliters per minute. So it's a lot. It goes pretty fast. That is the rate that the filtrate is produced at the Bowman's capsule. So that's how much fluid we're actually transferring. Now we can regulate glomerular filtration through a couple of ways. Why do we need to do it? First of all, because the reabsorption of the materials depends on the rate that the fluid is flowing. If you have too much filtrate, so if you have too much fluid going through the nephron at one time, it's going to be forced to move too fast because there's only so much space within the tubules of the nephron. So the fluid is going to have to move fast. While it's moving fast, you are not going to have enough time to reabsorb the good stuff. So you're going to risk losing things that you would or ultimately want to keep. You're going to end up losing them in the urine. You're going to pee them on out. However, if you don't have enough filtrate, so the filtration rate is too slow, then the fluid is going to be moving slowly through the nephron, which means that just about everything is going to get reabsorbed because you have a lot of time that is going to be slower moving through. So that means including some of the bad stuff that needs to be cleared and eliminated from the body is actually going to get absorbed back into the bloodstream when you don't really want it to. Turn the page. So what we do, our bodies do, is they're going to regulate the amount of filtrate that's produced, so how fast things are going to go through the nephron tubules, by altering, this is important, the diameter of the afferent, A-F-F-E-R-E-N-T, afferent arteriole. That would be the one coming into the glomerulus. The efferent is the one going out. We don't do anything with the efferent. Everything happens to the afferent arteriole. Write that down, put a star by it, whatever. Now, so if we increase the diameter of the afferent arteriole, we are going to increase the blood flow into the glomerulus, which would increase the blood pressure in the glomerulus. So then you're going to have more hydrostatic pressure. So fluid's going to move from high pressure to low pressure. So it's going to move into the glomerulus, resulting in an increase in GFR. So you have more fluid. On the other side, if you decrease the diameter of the afferent arteriole, you are going to decrease the blood flow into the glomerulus, which will decrease the blood pressure, which will decrease GFR. So everything either is increased across the board or it's decreased across the board. It doesn't get confusing in that regard. All right, there are intrinsic controls, also known as renal autoregulation. So that's something that the kidney does itself. So the kidney can try to maintain a constant blood pressure within itself despite the changes that you may be experiencing throughout the rest of the body with regards to your blood pressure. So the first one is the myogenic mechanism. So myogenic means muscle generated, and that's taking advantage of the natural tendency of smooth muscle where it wants to contract in response to stretching. So an increase in blood flow or blood pressure in the afferent arteriole leads to an increase in GFR, which is going to cause it to stretch. The afferent arteriole is going to stretch. Then, after it stretches, what's it going to naturally want to do? It's going to want to contract or release. So, after it stretches, it's going to contract in response, which is going to decrease the blood flow to the glomerulus, which is then going to decrease GFR. So it's going to be increased, and then it's naturally going to decrease after it's responding to stretching. There's also a function called the tubuloglomerular feedback mechanism. All right, this happens at, remember we talked about this in the lab, the juxtaglomerular apparatus. You can just call it JG, okay? And that happens where the distal convoluted tubule, so that's going to be nearing the end portion of the nephron, it twists back and it actually makes contact on the glomerulus with parts of the afferent arteriole. Now, the macula densa cells are going to be the cells that are part of this in the distal convoluted tubule, so the part that's actually on the nephron that has the macula densa cells. And they're going to then touch the cells of the JG apparatus in the afferent arteriole. Afferent arteriole, don't forget that. Now, if there is an increase in GFR, meaning you've got too much filtrate, so things are moving too fast, or an increase in concentration of sodium. So sodium is also something that will drive this. But ultimately, you're talking about fluid moving too fast. So fluid's moving too fast, that means in the distal tubule, it's going to be stretched a little bit. So that's going to communicate that through these macula densa cells to the JG apparatus on the afferent arteriole. And they're going to say, slow down please. So that's going to cause constriction within the afferent arteriole. When the arteriole is constricted, you have a decrease in blood flow to the glomerulus, which means you have decreased blood pressure in the glomerulus. So there's less hydrostatic pressure, so you don't get as much fluid, you have a decrease in GFR. This is a good thing. Now, if you have a drop in GFR, meaning you have too little filtrate, so things are moving too slowly and you're reabsorbing things that you no longer need, these cells, macula densa cells in the distal convoluted tubule, are going to communicate with the JG cells in the afferent arteriole and say, please vasodilate, we need more. So you're going to have vasodilation in the afferent arteriole, which is going to cause an increase in blood flow, which is going to cause an increase in blood pressure in the glomerulus, so you have a higher hydrostatic pressure difference, more fluid, increase in GFR. This is stuff that is happening multiple times, probably even within a second. All over the kidney. It's just a normal part of the process. You don't even think about it. The kidney does it itself. You can also manipulate it extrinsically. So if you will look on page 961, figure 24.14, 24.14. All right. Extrinsic controls. You can be controlled neurally or hormonally in addition to what the kidney does itself. First of all, the sympathetic nervous system. So we're talking about fight or flight. Sympathetic nervous system will cause vasoconstriction of which vessel? The afferent arteriole. That's the only one we're going to manipulate. So that's going to decrease blood flow to the kidneys. And why is that a good thing? One, because you don't want to have to pee when you're running away. And second, more blood flow is available for skeletal muscle. There's also the renin-angiotensin mechanism. So we've talked about this in every single unit we've done. So the JG cells in the afferent arteriole, they're going to detect a decrease, that's the arrow there. When they detect a decrease, they release the renin. Renin serves to convert angiotensinogen to angiotensin II. Angiotensin II causes an increased release of aldosterone from the adrenal cortex, which causes an increase in sodium reabsorption, which means an increase in water retention because water follows sodium. So ultimately you have an increase in blood volume and an increase in blood pressure. On the other hand, high blood pressure activates baroreceptors in the heart, in the atria, particularly the right atrium. And they're going to release atrial natriuretic peptide. We've already talked about this one before, ANP. And it is going to cause vasodilation, or vasodilates. It's going to be which vessel? The afferent arteriole. So that's going to increase GFR, increase the amount of filtrate, increase the amount of urine. When you increase the amount of urine, you decrease blood volume and decrease blood pressure. This is a good thing. Now, so this is like our natural diuretic of sorts. Now, if systemic blood pressure drops below 50 millimeters of mercury, no matter what happens, so that'd be your top number on your blood pressure, drops below 50, there is no filtration. You cannot establish a hydrostatic pressure, and so you get what is called anuria, which means without urine. And if that happens, anuria is technically considered a urine output that's anything less than 50 milliliters per day. 50 milliliters for 24 hours is not a lot. Anuria can also happen as a result of just renal failure. So people who are on dialysis, they're not going to be producing urine. They may occasionally have to empty their bladder once every few days. But they're not generally actually producing urine from their kidneys. But ultimately, remembering if systemic blood pressure gets too low, that you're not going to be able to establish any GFR. So that's filtration. Next, we're going to talk about tubular reabsorption. So this is going to be happening in the proximal convoluted tubule and the loop of Henle. Maybe to a little bit of an extent, the distal convoluted tubule. All right, so in tubular reabsorption, let's fill in our blanks. Materials are passing from the nephron tubules, that's the first blank, so from the tubules in the nephron, to the peritubular capillaries. Those are the blood vessels that are wrapped all around the nephron. It starts as soon as the blood gets through the glomerulus and enters the nephron in the proximal convoluted tubule, continues to the distal convoluted tubule. So as soon as stuff gets in there, the body's going to start grabbing back things that it wants to keep. Now the tubule cells are joined by tight junctions, so they are real tight. So there is a very controlled movement of what's allowed to pass. Now, what do you reabsorb? Generally, you want to reabsorb good stuff, things you want to keep in the blood. You don't want to get rid of good things. All right, organic nutrients. So you're talking about glucose and amino acids. Generally, there's not fats, but organic nutrients, fuels. You want to retain enough water and ions to maintain body homeostasis. So you want to make sure you have a good balance of sodium and potassium and water, particularly. Depending on the material, the reabsorption may be passive or it could be active. Now, first thing let's talk about active reabsorption. All right, active reabsorption is where things move up the concentration gradient or the electrical gradient if you're talking about an ion. Active reabsorption requires energy, so it does use ATP. It's limited because you only have so many transporters. You know, the boats can get full. Once the boats get full, even though there may be more in the filtrate, you can't pick it back up into your blood, so there is a saturation point. This happens with patients who have diabetes. If their blood glucose gets anywhere more than 180, that's the limit of the transporters in the kidneys. So blood sugars greater than 180, you're going to start to see glucose in the urine. Way back when, before we got good at testing blood glucose with the monitors and having point of site testing, patients would actually have to test their urine. It's like, well, if there's no glucose in the urine, then we know it's less than 180, but we didn't always know what it was. So, but that was how they used to have to check, was through the urine. You can still actually buy the urine glucose testing strips if you want them, but we don't really have much need for them. Active reabsorption is very specific. So you have one transporter for one type of what you want to keep. Things that are actively reabsorbed. So we want to keep them so much that we are willing to expend energy to keep them. Glucose, amino acids, sodium, vitamins, and most of our ions. So good things we want to keep. Passive reabsorption does not require energy. So it's things like diffusion or facilitated diffusion and osmosis. So sometimes there's some gated channels there. Substances are moving down a concentration or an electrical gradient. Does not require energy. No ATP is required. Here you're talking about obligatory water reabsorption. So water follows sodium, remember. So if we're actively reabsorbing sodium, we're going to passively reabsorb water. You do not have to actively reabsorb water. It happens as a result of water following sodium. There is a concept called solvent drag. So water is following sodium. The water could potentially have things dissolved in it that you don't necessarily want to keep. So some of the solutes that are in the water, as it gets reabsorbed, can get dragged along. Examples here are urea, which is a waste product that you do not need. You want to get rid of, but some of it ultimately gets reabsorbed as a result of solvent drag. Some toxins work this way. I don't have any specific examples, but they could. Now, materials that are not reabsorbed. These are going to be your nitrogenous wastes. So the end products that are of protein and nucleic acid metabolism, things you do not want reabsorbed, generally. Urea from amino acid breakdown, again, unless we're talking about solvent drag. Creatinine, that's from creatine phosphate breakdown, so we're talking about muscle tissue. So creatinine, if you look at renal function in the charts, it'll say estimated creatinine clearance, which is CrCl, which is abbreviated CrCl. That's what we'll use to measure clinically GFR. Uric acid from nucleic acid breakdown. If there's an issue with uric acid buildup, that's what we call gout. So excess uric acid is gout. All right. The proximal convoluted tubule, you can abbreviate it PCT. That's going to be the most active site of reabsorption. Reabsorption can also be determined or regulated hormonally. We've already talked about a lot of these, but let's mention them specifically within the context of how they are working in the nephron. Aldosterone. What does that aldosterone do? Aldosterone is a mineral corticoid, so it comes from the adrenal cortex, and it regulates the reabsorption of sodium. Causes the reabsorption of sodium, which causes, as a result, increased reabsorption of water, because water follows sodium. So we're going to get a net increase in blood volume to increase blood pressure. ADH, antidiuretic hormone. So diuretics make you pee, so antidiuretics make you not pee. It regulates the reabsorption of water, and it is released from the posterior pituitary. So in our little flow chart, it is going to increase the reabsorption of water directly without regard to sodium. And so that's going to cause an increase in blood volume, which will cause an increase in blood pressure. Vasopressin is what is synthetic ADH that you may see in the chart or give on the floor. All right, Pth Parathyroid Hormone is going to regulate the reabsorption of calcium. It's released from the parathyroid glands, and it causes an increased reabsorption of calcium. No water has to be involved there. Turn the page. Last part we're going to talk about, tubular secretion. This is where materials are passing the other way. They're going from the blood in the peritubular capillaries back into the nephron tubules so that they can get excreted in the urine. So they're going to move in your blank, move from peritubular capillaries to the distal convoluted tubule. This happens at the very end of the nephron, and to some extent it can even be secreted into the collecting ducts at the very end of the nephron. So what stuff gets secreted? Things are bad stuff. Bad stuff gets secreted, things you do not want. So drugs, penicillin is a good example. I don't know if you've ever noticed the odor of urine in a patient who's taking penicillin or amoxicillin. Has a very distinct odor because it is actively secreted. So it's one of the reasons it has a decreased half-life. You do have to take it more often. You can secrete things that get accidentally reabsorbed through solvent drag, so things that you accidentally reabsorb through because of the water. A big example of that is urea. Urea is very soluble, so it can get picked back up with the water, but then you get further down the nephron and it gets pushed on out. Your body will excrete enough potassium to maintain electrolyte balance. Remember, we're reabsorbing sodium. We tend to excrete potassium, but we can keep a little bit of potassium. We can secrete potassium to keep things balanced. Remember, too much potassium can cause serious cardiac arrhythmias, so we don't want to let it get too high. All right, tubular secretion can be involved to control the pH of the blood. Now remember, we're talking about pH. This is a direct response of the carbon dioxide bicarbonate equation that we talked about in respiratory. It comes back. Now, if you have low blood pH, meaning your blood is acidic, you have too many hydrogen ions, right? That means you're acidic. So in the nephron, you can actually secrete hydrogen ions and you can secrete bicarbonate ions. So remember our equation, CO2 plus H2O with carbonic anhydrase forms carbonic acid, which then dissociates into the bicarbonate ion, H2CO3, plus the free hydrogen ion. So here we're talking about the stuff on the right-hand side, the free hydrogen ion and the bicarbonate ion. So if our blood is too acidic, we have too many hydrogens, free hydrogens. The body, the blood, is going to increase the secretion of the hydrogen ions from the blood into the urine, and it's going to decrease the amount that it secretes of bicarbonate. It may still secrete a little bit, but it's going to decrease how much it's actually secreting bicarbonate. On the other hand, if you have a high blood pH, meaning blood is alkaline, you have too few H pluses in the blood, so you don't have enough free hydrogen in the blood. In that case, the body will decrease the secretion of hydrogen, and it will increase the secretion of bicarb, or the bicarbonate ion there. So depending on the pH of the blood, you can increase or decrease how much you secrete of hydrogen ions versus bicarbonate ions. Now, as a general rule, why do we care? You want to retain the bicarbonate ion in the blood so your free hydrogen ions can recombine, and ultimately, when you get to the lungs, exhale that as CO2. So you do want to regulate how much bicarb you have So you could, because the way we ultimately get rid of it is exhaling it through the lungs. We can secrete it through the urine, but it's not near as an efficient mechanism as exhaling. Now, let's talk about urine. Once things get to the end in the collecting duct, you have formed urine, so then it's just going to follow the process, out of the body. Urine color. It tends to be deep or dark yellow. That is from a substance called urochrome. urochrome, urochrome. That comes from hemoglobin breakdown into bilirubin to make bile. So breakdown of bilirubin there. Sometimes urine is cloudy. That's generally due to a urinary tract infection. Urine odor can have an ammonia smell. That happens from the breakdown of amino acids. Amino acids are broken down into urea. Urea can also be further broken down into ammonia. So you can get the characteristic ammonia smell. I already talked about penicillin, which is actively secreted. It does have its own unique odor as well. You can get a fruity smell, potentially, from a patient with diabetes mellitus. And that happens not only as a result of glucose overflow in the urine, but potentially when the glucose has been elevated for a significant enough period of time that they've gone into a ketotic state. So the ketones, which, you can have the fruity smelling breath in the ketotic, when they get in ketoacidosis. But even before they get to full-on ketoacidosis, they'll be excreting the ketones in the urine, which can give it that kind of smell. There are people, that will actually test their urine for ketones. These are people that generally Atkins diet are low-carb people that want to be using fats and proteins for energy versus carbohydrates. So they actually try to be in a ketotic state. So that's actually one of the things that they'll test for, and you can get urine testing strips for ketones. Keto sticks is what they're called. You can purchase them. Now, normal pH of the urine is somewhere around 6. So if you think about what's normal blood pH, 7.35 to 7.45. So urine is more acidic than blood. Typically, it should be. And that happens because when we were talking about our secretion with regards to hydrogen ions and bicarbonate ions, ultimately more hydrogen ions get secreted than bicarbonate, even when you're trying to balance those. So ultimately you're going to get H plus secretion, which results in urine that's going to be more acidic than the blood. That's done on purpose. Now, things that you should not find in the urine. These are bad things. And I'll tell you what they mean. Glucose in the urine, indication of diabetes mellitus. Ketones in the urine, indications of starvation. So you're talking about the body acting like there's starvation. So excess breakdowns of fats and proteins. Again, there are people on the Atkins diet, try to be in a ketotic state. It doesn't necessarily have to be Atkins diet. Ketogenic diets are used sometimes in children to help control seizures or other types of, behavior. Those are options. So sometimes it's because of actual starvation, sometimes it's because of simulated starvation because of dietary choices. Proteins should not be in the urine because proteins as a rule are too big to fit through the glomerulus, through those fenestrated capillaries, and through the filtration slits on the podocytes. So if you have proteins that are slipping through, that is going to damage, and generally is irreversible. It can cause irreversible damage to the glomerulus. Now you have lots of nephrons, so you have lots of glomeruli, but over time, that is a major cause of a decrease in kidney function, ultimately to renal failure. That's one of the things we monitor for in people with diabetes is protein in the urine, albuminuria. Should not have it there. Bile pigments should not be found in the urine. The liver should be breaking down bile pigments. I'm not talking about urochrome here. That's from actual bile acid breakdown, so that's different. You should not be finding erythrocytes, no red blood cells in the urine. They're too big to be filtered. If they're there, that's bad. Blood in the urine is not a good thing. Hemoglobin should not be found in the urine. It should not only stay inside red blood cells, but it is also a protein. So even if hemoglobin is out of the red blood cells, it still should not be getting through the glomerulus. Leukocytes, white blood cells should not be in the urine. If they are, is an indication of an infection. Nitrites are also something we test for in a UA that can be an indication of a urinary tract infection. Turn the page. The last page is the parts of the kidney nephron that we covered in lab. And that concludes Chapter 24, the urinary tract.